Detecting the Earth's magnetic field presents a biophysical paradox. The force is weak, falling far below the threshold necessary to trigger standard bioelectric induction. The Earth's magnetic field sits at approximately 0.05 microteslas. For a flying bird, the induced electric field is less than 1 millivolt per meter, significantly lower than the energy required to fire a standard action potential. Because the geomagnetic field is static and weak, a classical thermodynamic sensory mechanism would struggle to function. The ambient thermal noise of the bird's own body would likely overwhelm a signal of this magnitude. Since classical thermal mechanisms are too coarse for a signal this faint, the avian compass requires a level of sensitivity found in quantum kinetic processes. This quantum detection occurs within the photoreceptor layer of the avian retina. The molecule responsible for this transduction is a light-sensitive protein called cryptochrome 4, or CRY4. Cry4 molecules are not distributed at random. As this diagram shows, they are anchored within stacked membrane disks inside the retinal cone, maintaining a highly ordered, grid-like alignment. Unlike other cryptochromes used in biological clocks, Cry4 possesses a stoichiometric complement of an FAD cofactor, a specific molecule that allows it to absorb light. This combination of a rigid molecular scaffold and a light-absorbing cofactor provides the necessary framework to act as a quantum sensor. When blue light strikes the FAD cofactor, an electron transfer creates a radical pair. These oscillate between a singlet state, which allows recombination, and a triplet state, which forces Cry4 into an active signaling state. Internal magnetic moments drive this rapid interconversion. The biochemical output of the protein is determined by the ratio of time the pair spends in the singlet state compared to the triplet state. This internal quantum oscillation is where the Earth's magnetic field exerts its influence. Through a principle called Zeeman splitting, an external magnetic field causes the single triplet state to split into 3 divergent energy levels. This widens the gap between the singlet state and 2 triplet states, blocking interconversion and shifting the singlet to triplet ratio. The severity of this split is highly dependent on the physical angle of the magnetic field vector relative to the radical pair inside the protein. By altering these subatomic spin states, the magnetic field translates an invisible directional angle into a tangible chemical signal. Scaling up from the molecule to the macroscopic, we look at the hemispherical geometry of the avian eye. Because avian cone cells tile the entire retinal surface, the Earth's magnetic field lines intersect different clusters of receptors at vastly different angles. These varying angles generate a topographical gradient of radical pair yields. This creates a chemical heat map across the retina that shifts as the bird moves its head. This gradient effectively projects a visual overlay of the magnetic field directly onto the bird's field of view. Retinal ganglion cells then convert this analog chemical map into digital action potentials. Subatomic physics has now been scaled into a digitized cellular map traveling down the optic nerve. The brain must now process this magnetic data without interfering with the bird's standard vision. Image-forming vision is typically routed through the tectofugal pathway. However, the magnetic compass data is segregated. It is routed through a different circuit entirely, the thalamofugal pathway, which leads directly to the dorsal forebrain. This specialized routing isolates the magnetic signal, allowing the brain to process navigational data independently from visual objects. The thalamofugal pathway terminates in the visual wulst of the forebrain, in a region known as cluster N. These cross-sections track neural activity via zinc expression. The day-active sample on the left shows little activity, while the night-active sample on the right is filled with dense, darkened dots. The data shows that cluster N undergoes significant bilateral neural activation exclusively at night. This nighttime activation correlates with Sugenru, the specific behavioral state of migratory restlessness. This 3D reconstruction shows the physical extent of cluster N in the forebrain, where it envelops the dorsal nucleus of the hyperpallium. Cluster N serves as the executive processor that translates the retinal magnetic image into a navigational heading. Contrasting this system with mammals highlights the unique biological requirements of the avian compass. While mammals possess cryptochromes, type 2 cry in mammals lacks the stoichiometric FAD cofactor. It functions as a gear in the circadian clock, rather than a quantum sensor. The Avian compass relies on a specific sequence, beginning with a subatomic vulnerability to Zeeman splitting inside the CRY4 protein. This is followed by the macroscopic mapping of that reaction across a hemispherical retina. Finally, this data is digitized and routed into the specialized cluster N processor. The Avian magnetic compass operates through a sequence that spans from subatomic quantum mechanics to cellular topography and systems neurobiology.